HighBridge, a division of Recorded Books, presents The Catalyst, RNA and the Quest to Unlock Life's Deepest Secrets, by Thomas R. Cech. Read by Joshua Saxon. Publisher's note: in this audio edition, you will hear references to visual and coding sequences present in the print and ebook formats of the book. Introduction. The Age of RNA. It is often said that the first half of the 20th century was the age of physics. The curvature of spacetime, the dynamics of subatomic particles, the Big Bang and black holes, the unleashing of atomic energy that could power whole cities or obliterate them. All these discoveries burst forth to revolutionize science and change our everyday lives. One could say it was the Big Bang of physics itself, taking place roughly between 1905, when Einstein gave us E equals mc squared, and 1947, when the transistor was invented at Bell Labs. As the second half of the 20th century dawned, biology began to nudge physics out of the scientific spotlight. And by biology, I mean DNA. After all, that half-century more or less began with Francis Crick and James Watson's momentous discovery of the DNA double helix in 1953, and it ended with the Human Genome Project from 1990 to 2003, which decoded all of our DNA into a biological blueprint of humankind. Today, everyone knows DNA's greatest hits, that it contains our genetic information, that it can be used to trace our ancestry and pinpoint hereditary diseases and solve crimes. DNA has even entered the vernacular. If I tell you that something is in my DNA, whether it's a devotion to hiking in the mountains or a love of Thai food, I'm saying that it's a core part of who I am, my essence. During the age of DNA, RNA was mostly overlooked by the general public. Certainly textbooks described and students learned how RNA, the ribonucleic acid to DNA's deoxyribonucleic acid, was copied from the double helix, and how messenger RNA, or mRNA, worked to transmit DNA's code to instruct the synthesis of proteins. But RNA was never the star of the show. It was like a biochemical backup singer, slaving away in the shadow of the diva. Yet, to those in the scientific community, RNA's previously unappreciated talents began to become apparent. RNA is tiny, measuring only about a nanometer in diameter. If you stacked molecules of messenger RNA side by side, you could fit 50,000 of them within the breadth of a single human hair. Yet, as researchers started to discover what RNA lacks in size, it makes up for in versatility. Folding into origami-like shapes, it can pull off wild stunts that make its genetic parent DNA look like a one-trick pony. In fact, DNA has just one trick, albeit one that's central to all life on Earth. DNA stores genetic information. That's it. It's like the hieroglyphics in an Egyptian mummy's tomb, or the grooves in a vinyl LP record, or the ones and zeros that make up the bits of information stored in a computer. DNA's job is to sit there in the cell nucleus, storing information. Reading out that information and doing something with it requires proteins and RNA. The first thing to understand about RNA, then, is that it is a many-splendored thing. Yes, it can store information just like DNA. As a case in point, many of the viruses that plague us don't bother with DNA at all. Their genes are made of RNA, which suits them perfectly well. But storing information is only the first chapter in RNA's playbook. Unlike DNA, RNA plays numerous active roles in living cells. It can act as an enzyme, splicing and dicing other RNA molecules or assembling proteins, the stuff of which all life is built, from amino acid building blocks. It keeps stem cells active and forestalls the aging process by building out the DNA at the ends of our chromosomes. By guiding the gene-editing machinery of CRISPR, it empowers us to rewrite the code of life. Many scientists believe RNA even holds the secret of how life on our planet began. At last, RNA has begun to step out of DNA's shadow to reveal its own immense potential. Since 2000, RNA-related breakthroughs have led to 11 Nobel Prizes. In that same period, the number of scientific journal articles and the number of patents generated annually by RNA research have each quadrupled. There are more than 400 RNA-based drugs in some stage of development, beyond the ones that are already in use. And in 2022 alone, more than $1 billion in private equity funds was invested in biotechnology startups to explore new frontiers in RNA research. While DNA may have dominated biology research in the past, RNA has clearly become the focus of the future. The 21st century is already standing out as the age of RNA, and this century still has a long way to go. This book is an educated citizen's guide to understanding how RNA, literally and metaphorically, went viral, how it transformed from an arcane topic mostly of interest to biochemists to a mainstream subject that is shaping the future of science and medicine. I come to the story not as an impartial observer, but as an active participant. As a chemistry and biochemistry professor at the University of Colorado in Boulder, I have been studying RNA for most of my career. I've been an eyewitness to discoveries about RNA that caused scientists to rethink the deep question of how life originated on planet Earth and revealed breathtaking insights about human health and disease. A few of these discoveries my research group and I made ourselves. Others were the work of close friends and colleagues, and so it only feels right to refer to them by their first names. Taken together, these breakthroughs in RNA research represent one of the most transformative scientific achievements since the discovery of the DNA double helix. Yet for many years the public was not well equipped to appreciate this accomplishment because people had only a hazy idea of what RNA is or why scientists were so excited about it. I always thought this was a shame, for the stories are thrilling. In addition, the public financed much of this research with its tax dollars, and people deserve to know how their investment has paid off. Then, in the tumultuous spring of 2020, the public came face to face with RNA in a striking way. My work, like that of so many others, had for the moment come to a halt. My lab was shut down, my classes were canceled. But my subject was suddenly on the tip of everyone's tongue. The world was being ravaged by SARS-CoV-2, the RNA-based virus that causes COVID-19. To combat the virus, mRNA vaccines were being developed with unprecedented speed, a stunning achievement built on decades of fundamental research breakthroughs in RNA science. Breakthroughs about which most people knew nothing at all. Naturally, the public wanted to understand this molecule that was both the cause of and the potential cure for our troubled times. So I went from RNA scientist to RNA spokesperson. I made it my mission to demystify RNA, first in public talks and now with the book you hold in your hands. I tell the story of RNA in two parts. The first is the story of how RNA revealed itself as life's great catalyst. We begin in the 1950s with the experiments that uncovered how RNA orchestrates the construction of the proteins that perform most of the essential functions in living organisms, from holding cells together to metabolizing food. Then we see how RNA, through a curious transformation called splicing, is responsible for helping us humans perform so much more with our DNA information than, say, a fungus, a worm, or a fly can. From there, the story takes a personal turn. I recount how my team discovered catalytic RNAs called ribozymes, whose existence violated what had been considered to be a bedrock rule of nature, that enzymes must be proteins. This breakthrough led to the 1989 Nobel Prize in Chemistry and marked a major turning point in RNA's story. The moment when the world of science started to see this molecule not as a passive messenger, a bit player in the chemistry of life, but as the star of the show. The next major challenge was to map the wondrous shapes that RNA assumes to accomplish its multiple miracles, an effort that had humbled even the great James Watson after his success solving the structure of DNA. We then observe how RNA was discovered to be the secret power source behind the ribosome, the mothership in our cells that reads the code contained in messenger RNAs and then uses it to construct the proteins that power so much of life. Finally, we examine how RNA could answer the greatest chicken or egg problem in science, how life on Earth began almost 4 billion years ago. While the first part of the book describes how RNA supports life, the second part shows how RNA can improve and extend life beyond nature's current limits. We start with the extraordinary story of telomerase, an RNA-powered enzyme that has taught us that immortality and cancer are really flip sides of the same coin. Next, we learn how tiny RNAs that work like switches, turning off messenger RNAs in cells, are being repurposed to short-circuit disease. But RNA doesn't only cure us, it also can kill us. RNA is the genetic material of many of the deadliest viruses in history, from polio to SARS-CoV-2. While these viruses make RNA into a villain, mRNA vaccines show us how RNA can still save the day, protecting us not only from COVID-19 but also, perhaps, from cancer and many other diseases. And at last, RNA gets its ultimate revenge on DNA as the power behind CRISPR, a system that gives us the ability to remodel DNA itself. CRISPR has already revolutionized fundamental scientific research, and profound applications in medicine and in slowing the march of climate change are almost upon us. As it turns out, the same versatility that enables so many vital functions in nature also makes RNA the perfect tool for biomedical engineers who are now redefining life as we know it. Given that the hero of this book is present in every living thing on the planet and has been around for about 4 billion years, there was no way I could tell the whole unabridged story of RNA. I had to make hard choices about what to include and what to leave out. I also had to simplify many of the scientific concepts to make them more accessible. At times, I describe RNA as spaghetti and compare RNA splicing reactions to copying and pasting text in a word processing program. Such simplification may irritate my colleagues, but as my wife and fellow biochemist often reminds me, I am not writing this book for them. I expect I'll annoy my science friends in yet another way. In telling this tale, I have tried to keep the spotlight on RNA itself. So while my stories feature some of the researchers who stumbled onto key discoveries, or who took several wrong turns before uncovering the truth, I make no claims at being comprehensive. For each scientific topic covered in this book, I have tried to credit additional researchers in the endnotes, although I apologize in advance for the multiple offenses of omission I've likely committed. One of the wonderful things about today's RNA science is that most discoveries have multiple contributors who are constantly passing the ball to each other, as in a game of rugby. Also, as in rugby, the researchers occasionally pile into a ruck, where for a while the ball seems lost. It's a competitive, messy, at times painful game, but one that is punctuated by moments of glory. Part 1. The Search. Chapter 1. The Messenger. George Gamow had solved many imposing scientific problems before he turned his attention to cracking the code of life. Born in 1904 in the Black Sea port of Odessa, Gamow began contemplating the universe at age 6 when he saw Halley's Comet from the rooftop of his family's apartment building. 4 decades later, he would become the world's leading proponent of the theory that the universe began with a Big Bang. Gamow's fellow scientists regarded him as a genius, another Heisenberg, said Niels Bohr, comparing him to the Nobel Prize-winning pioneer of quantum mechanics, but also an oddball, a giant imp jumping from atoms to genes to space travel, said Jim Watson. Gamow stood out for his 6'6" height and for his puckish sense of humor, displayed even in the most serious academic settings. When publishing the theory of the cosmological origin of the chemical elements he had developed with his student Ralph Alpher, Gamow popped in the name of his colleague Hans Bethe, simply to create an Alpha Beta Gamma author list befitting the Greek alphabet. After defecting from the Soviet Union in 1933, Gamow arrived in the United States a year later. A professor of physics for 20 years at George Washington University in Washington, D.C., Gamow eventually made his way to my own University of Colorado in Boulder, where the tallest building on campus, the Gamow Tower, housing the physics department, is named in his honor. Though he had cut his teeth on nuclear physics and cosmology, Gamow became convinced by the early 1950s that the most exhilarating scientific question left to answer had nothing to do with the origin of the universe or the behavior of subatomic particles. In fact, it had nothing to do with physics at all. In June 1953, Gamow read Jim Watson and Francis Crick's momentous article in the journal Nature announcing that the structure of DNA was a double helix. The structure provided a neat solution to what had been a major mystery: how genetic information is duplicated so it can be passed from one generation to the next. The 4 chemical units, or bases arranged along a DNA strand, adenine A, thymine T, guanine G, and cytosine C, form base pairs with complementary bases on the other strand of the double helix. A always matches with T, and G always matches with C. The double helix looks like a twisted zipper. If one were to unzip the zipper, each side would have all the information needed to direct the construction of the opposite side, as the 2 sides are always perfectly complementary. This must be how genetic information is replicated, Watson and Crick reasoned. In a letter dated July 8, 1953, Gamow congratulated Watson and Crick for bringing biology into the exact sciences. He boldly proposed a collaboration across disciplines to tackle the next big question that was on everyone's mind: how the information encoded in those strings of A, T, G, and C was actually read out to specify ultimately a hand, a heart, a liver, or a brain, or the wrinkled and smooth peas in the garden of Gregor Mendel, the Augustinian monk who first proposed that such traits were passed down from generation to generation in the form of some elementary unit, which we now call a gene. Gamow suggested that he could help Watson and Crick use math and physics to decrypt this genetic code. Watson and Crick were flattered that a famous physicist was interested in their work, but Gamow's handwritten letter had so many whimsical qualities, Watson recalled, that we did not know how serious he might be. Gamow was very serious. Over the next several months, he threw himself into deciphering the genetic code. He was a consultant to the U.S. Navy at the time, so he sought out not only chemists and physicists to help him in his quest, but also military cryptographers. As it would turn out, however, solving this secret of DNA would ultimately require DNA's own daughter, ribonucleic acid, or RNA. A riddle wrapped in a necktie. In biological terms, deciphering the genetic code means understanding how DNA encodes proteins. Life is built from proteins, which are the main movers and shakers in every organism in our biosphere. In humans, some proteins form structures such as muscle fibers, skin, and hair. Some act as enzymes, breaking down the food we eat into its constituent components and then recycling these pieces to build up new cellular machines. Others punch holes in the envelopes that encase our cells, selectively allowing some salts or nutrients to enter the cell and expelling others. Other proteins act as signaling molecules, receiving information from the outside world and activating internal processes accordingly. Still others are antibodies, which protect us from foreign invaders such as viruses. In short, proteins are spectacularly diverse. Chemically speaking, proteins are polymers, strings of 100 or even 1,000 amino acids. These amino acid building blocks come in 20 types, with names such as lysine, valine, and phenylalanine. Each protein has a specific order or sequence of amino acids along its chain. This sequence determines how the protein folds into a three-dimensional entity that has a particular function, such as digesting food in the stomach or transmitting neuronal signals in the brain. Thus, even though we hear that we need to eat fish or tofu or an impossible burger to get enough protein in our diet, there is not a protein, but rather many thousands of different ones, and each is encoded by its own gene, which is made of DNA. Whereas proteins are built out of 20 amino acids, DNA is built out of the 4 bases. This leaves us with, on the one hand, the sequence of A's, G's, T's, and C's in a gene made of DNA, and, on the other hand, the sequence of 20 amino acids in the corresponding protein. Gamow sought to figure out how different arrangements of the 4 DNA bases specified, or encoded, each of the 20 amino acids. By 1954, he had recruited Watson, Crick, and an eclectic cadre of well-known scientists, 20 in all, 1 for each amino acid, to direct their brainpower to the coding problem. Gamow had woolen ties, each embroidered with a strand of RNA, sewn for the members of this little fraternity of pointy-headed code breakers. He dubbed the group the RNA Tie Club. Why not the DNA Tie Club, you may ask? It was Jim Watson who convinced Gamow that they had to focus their code-breaking attention on RNA because of the basic structure of cells. Whereas DNA is found inside the cell's nucleus in higher organisms, proteins are made outside the nucleus in the region known as the cytoplasm. This spatial separation mandated that some sort of messenger must transport the information from DNA to the site of protein production. A number of forward-thinking scientists thought that RNA could be that messenger, as it was known to be abundant both in the nucleus and in the cytoplasm. Since the early 1900s, chemists had analyzed RNA and DNA and found that they were cut from the same biochemical cloth. Their full names, deoxyribonucleic acid and ribonucleic acid, reveal how closely related they are. Deoxy indicates that DNA has 1 fewer oxygen atom on each of its repeating units than RNA. The extra oxygen atom makes RNA chemically much less stable than DNA. For many decades, DNA and RNA were seen as chemical compounds without a function. They were far less interesting to scientists than proteins, whose roles as enzymes and as signaling molecules such as insulin seemed so exciting. DNA emerged as the centerpiece of biology only when the 1944 discovery by Oswald Avery and colleagues at the Rockefeller Institute that DNA is the molecule responsible for causing heritable changes in bacteria was coupled with the 1953 discovery by Watson and Crick of the double helical structure of DNA. Because scientists had already theorized as early as 1947 that RNA was copied from DNA, they now assumed that RNA must also have an important role to play in the chemistry of life. Although RNA is typically single-stranded and DNA is double helical, both speak the same language. Like DNA, RNA is written in a four-letter alphabet. The first three letters, A, G, and C, are the same as DNA's. The fourth letter, U, or uracil, occupies the same place in RNA's alphabet as T does in DNA's alphabet. So when RNA is copied from DNA, it carries the same information. Devoted as always to big sweeping theories, Gamow first tried to work out a solution to the coding problem with nothing more than pencil, paper, and his brain. In 1953, the same year the double helix was discovered, he theorized that 3 bases might code for 1 amino acid. His reasoning was mathematical. If you took the 4 letters in the DNA or RNA alphabet and tried to create as many combinations of 2 letters as possible, you would get only 16 combinations, too few to specify the 20 amino acids that make up proteins. But if you tried to arrange the 4 letters of the DNA or RNA alphabet into as many 3-letter combinations as possible, you would get 64 of these triplet codons, enough to specify 20 amino acids and then some. Longer groups of bases could do the job, but 3 were enough. It was the most economical format for the genetic code. But which groups of 3 bases encoded which amino acids? Even though Gamow had brought together some of the greatest geniuses of the 20th century, ultimately the distinguished members of the RNA tie club succeeded only in tying themselves in knots. By the late 1950s, Gamow had given up trying to crack the code, convinced that the problem had no solution that could be found on the basis of pure theory. What Gamow really needed was the biological equivalent of the Rosetta Stone. Instead of a text written in both Egyptian hieroglyphics and Greek, it would display an amino acid sequence of a protein as well as the RNA sequence that encoded it. But there was no such stone. Scientists would need to devise their own, and to do it, they would have to determine whether this hypothetical messenger actually existed, whether RNA truly was the missing link between our genes and the protein building blocks of life. Did you get my message? In the 1950s, plenty of scientists, including Jim Watson and Francis Crick, fervently believed in the theory that RNA might carry information from the DNA in the cell's nucleus to the cytoplasm, where proteins were synthesized. But when scientists tried to find empirical evidence that RNA is the messenger of life, their initial results fell flat. Their first disappointment came when they found that most of the RNA in a cell's cytoplasm has the same proportion of A, G, C, and U bases independent of what proteins were being synthesized. This didn't make sense. It would be akin to finding that Beethoven's Ninth Symphony had exactly the same proportion of each kind of musical note as Lady Gaga's Bad Romance. You would expect that these very different pieces of music would have correspondingly dissimilar distributions of F sharps and B flats and so on. Similarly, you would expect that different proteins, with their different amino acid compositions, would have different proportions of A, G, C, and U in the messenger RNAs or mRNAs that specified them. The second disappointment was that most of the RNA in the cytoplasm is very stable. Once born, it has great longevity. But scientists had seen cases where the proteins being made in a cell switched rapidly within a few minutes from one set of proteins to a completely different set. For example, if you switch the food source for bacteria, they would stop making the enzymes they use to digest the old food and immediately start making enzymes suited to the new food. Likewise, if a bacterium became infected with a virus, bacteria are plagued by their own viruses called bacteriophage or phage, it would switch from producing bacterial proteins to producing phage proteins. Consequently, a true mRNA would need to be unstable to allow rapid changes in what proteins were being made. The rock-stable nature of most of the cellular RNA seemed to disqualify it from being the messenger the scientists sought. Among the scientists who remained convinced that there must be an mRNA hiding from view were François Jacob and Jacques Monod of the Pasteur Institute in Paris. They had made seminal discoveries of how genes were turned on and off in bacteria and were now turning their attention to RNA. In 1960, on a visit to Cambridge, England, Jacob met with his friend Francis Crick, Sidney Brenner, and others in Brenner's rooms at King's College. Jacob described his latest experiments on how genes were regulated in bacteria, and the conversation soon turned to excited speculation about the role of messenger RNA as the link between gene and protein. Suddenly, and almost simultaneously, Crick and Brenner jumped up they recalled recent experiments by two scientists at Oak Ridge National Laboratory in Tennessee, Ken Volken and Larry Astrachan. In studies of Escherichia coli infected by a phage called T2, Volken and Astrachan had witnessed the rapid formation of new RNAs that were smaller than the stable RNA in the cell, now known to be the RNA embedded in ribosomes, the cell's protein factories. For a host of complicated reasons, Volken and Astrachan had interpreted their data as indicating that RNA was metamorphosing into DNA. But what if instead they had caught a glimpse of the elusive mRNA? It was an exciting possibility, but a more direct test of the mRNA hypothesis would be necessary to confirm it. Sidney Brenner and Francois Jacob were set to visit the geneticist Matt Messelson at Caltech a few weeks later. It was decided that the three of them would perform the critical experiments. Messelson and his colleague Frank Stahl had recently used a new technique involving an extremely fast-spinning centrifuge to test Watson and Crick's conjecture that the strands of the double helix came apart during DNA replication. Their results supported the theory, finding that each progeny double helix retained one old strand from its parent and paired with one newly synthesized strand. Now, Brenner, Jacob, and Messelson would use this same ultracentrifuge to try to find the mRNA needles in the ribosomal RNA haystack. The plan was to infect E. coli bacteria with a phage, which would cause the bacteria to switch the type of protein they were producing. At the same time that the scientists added the phage, they would add a radioactive version of uracil, labeled with carbon-14, that would be incorporated only into newly made phage mRNA, not into pre-existing bacterial RNAs. If the mRNA hypothesis was correct, the new phage mRNA would appear at just this moment, allowing them to capture this missing link between DNA and protein. Indeed, Brenner, Jacob, and Messelson saw the radioactive phage mRNA in the ultracentrifuge, clearly smaller than the RNA of the ribosomes. As predicted, this RNA was short-lived, and it teamed up with the pre-existing ribosomes of the infected bacteria to produce the new kinds of proteins needed by the phage to do its dirty work. They had finally captured mRNA, as elusive as it was. One way of thinking about this biological process is to imagine a record player. The ribosome is your turntable, the mRNA is the vinyl LP record, and the protein is the music you hear when you lower the needle. You might change the record depending on your mood, but the rest of the setup remains the same. Just as a record player can play any LP record, ribosomes can work with any mRNA that comes along. The mRNA is what determines the specific protein produced, whether a phage protein, an E. coli protein, or something else, just as the record determines what music you're hearing. The reason mRNA was so difficult for scientists to detect is that only about 5% of the RNA in an E. coli bacterium is messenger RNA. The other 95% is dominated by ribosomal RNA. Moreover, E. coli has 4,000 different genes, each of which yields an mRNA of a different size and sequence. Thus, any given mRNA is far less than the 5% total. Finally, most E. coli mRNAs have a lifespan of merely a few minutes, making them elusive to capture. In contrast, ribosomal RNAs are not only omnipresent in the cytoplasm but also very long-lived. So it's easy to see why it took so long for pioneering scientists to see past all that ribosomal RNA. A linguistic interlude. Thinking about a record player might help us explain how ribosomes and mRNA team up to make proteins, but when it comes to understanding how RNA encodes proteins, we need to switch off the music and crack open a book. The pages of most books are filled with written language, letters, words, sentences. In the Book of Life, which is written in the language of DNA, we don't have an alphabet of 26 letters as in English or 22 letters as in Hebrew, but 4 letters, A, G, C, and T. The way those 4 letters are arranged, their sequence, will determine the meaning of each word and each sentence. So how many books do we need to capture the meaning of life, to capture all the DNA in a given organism? The answer depends, of course, on the size of the genome, a genome being the totality of the DNA in an organism. The human genome, bundled into 23 chromosomes, has about 3 billion bases, or letters. Assuming a typical font size that allows about 3,000 characters per page, it would take 1 million pages to record the human genome. Now, considering that a long book might have 500 pages, the genome sequence would require about 2,000 books. Too many for your home library, but just a fraction of what your local public library keeps on hand. Each human chromosome, comprising a single linear DNA molecule, would occupy about 90 books. A bacterial genome, such as that of E. coli, is a lot smaller.
Sample complete. Ready to continue?